Welcome to another episode from the Veterinary Medicine Digest. Today, we delve into a series of fascinating and critical studies published in Animal Genetics and Genes, focusing on a group of devastating inherited neurological conditions in dogs, specifically the Neuronal Ceroid Lipofuscinosis, or NCLs, and related neurodegenerative disorders. These conditions are characterized by progressive deterioration of cognitive and motor function, often leading to severe quality-of-life issues and premature death in affected animals. Our first paper, by Bellamy and colleagues, describes a case of Neuronal Ceroid Lipofuscinosis in a Schapendoes dog, highlighting a novel genetic variant and its implications. The broader field of lysosomal storage diseases, to which NCLs belong, represents a heterogeneous group of disorders where the body's cells fail to properly break down and recycle waste products, leading to their accumulation, particularly in neurons. This accumulation results in the characteristic neurodegenerative signs that we see in affected dogs, which often mirror those observed in human patients, making these canine models incredibly valuable for understanding disease mechanisms and exploring potential therapies. The research we're discussing today, conducted by various teams, aims to pinpoint the specific genetic mutations responsible for these debilitating conditions in different dog breeds, offering crucial insights into their pathogenesis and paving the way for improved diagnostic tools and breeding strategies to prevent future cases. The clinical presentation of these diseases can be varied, but typically involves a constellation of neurological deficits, often starting in early life and progressing relentlessly. Common signs include visual impairment, seizures, changes in behavior and cognitive abilities, and difficulties with movement and coordination. Pathologically, these disorders are often confirmed by the presence of abnormal intracellular accumulations of autofluorescent lipopigments and a significant loss of neurons within the central nervous system. These pathological hallmarks are central to diagnosing and understanding the specific forms of these neurodegenerative conditions.
Building on that, the Bellamy team's findings in the Schapendoes provide a clear example of how these genetic discoveries are made and their profound impact. They investigated a 19-month-old Schapendoes dog that presented with clinical signs highly suggestive of a lysosomal storage disease, which was subsequently confirmed by pathological examinations consistent with Neuronal Ceroid Lipofuscinosis. Specifically, the post-mortem examinations revealed typical histopathological findings, such as the accumulation of autofluorescent cytoplasmic inclusion material, which strongly supported their initial diagnosis. To uncover the genetic basis, whole genome sequencing was performed on the affected dog and both of its parents. This in-depth genetic analysis led to the identification of a missense variant in the CLN6 gene, designated as C.386T greater than C. This variant, located in a highly conserved region of the gene, was predicted to be harmful, thereby strongly supporting its role as the causal mutation for the observed neuronal ceroid lipofuscinosis in this dog. A crucial finding was that the affected dog was homozygous for this variant, meaning it inherited a copy of the mutated gene from each parent, while both parents were found to be heterozygotes, carrying one normal copy and one mutated copy of the gene. This pattern of inheritance is classic for autosomal recessive diseases, where an individual must inherit two copies of the faulty gene to express the disease. Furthermore, none of the healthy littermates of the affected dog were homozygous for this specific variant, reinforcing the correlation between the genotype and the severe disease phenotype. The clinical progression in this Schapendoes was tragically rapid. Signs first appeared at 12 months of age, and the dog was euthanized just 7 months later, at 19 months old, due to the severity of the neurological decline. This timeframe and the clinical signs, including notable fly-snapping behavior, were remarkably similar to those previously described for CLN6 affected Australian Shepherds, suggesting a common disease mechanism across breeds. The identification of this specific and novel CLN6 variant is incredibly significant because it enables the development of pre-breeding DNA testing. Such testing can prevent future cases of Neuronal Ceroid Lipofuscinosis type 6 in the Schapendoes breed by allowing breeders to identify carriers and make informed mating decisions, thereby reducing the incidence of this devastating inherited disorder. Moreover, this specific case in the Schapendoes dog presents a valuable potential natural model for human Neuronal Ceroid Lipofuscinosis type 6. Neuronal Ceroid Lipofuscinosis type 6 in humans is a debilitating, currently incurable, and lethal disease that primarily affects young children, with several hundred cases reported worldwide. The shared pathogenesis between dogs and humans with this condition underscores the importance of studying these canine models, as they can provide critical insights into disease progression and aid in the development of potential human treatment strategies. This particular Schapendoes case is notable as it marks the second dog breed where neuronal ceroid lipofuscinosis type 6 has been reported, further strengthening its utility as a comparative model.
That's an excellent point about the value of these canine models, particularly when they mirror human conditions. Now, let's shift our focus to another significant study by Reitman and colleagues, published also in Animal Genetics, which delves into neuronal ceroid lipofuscinosis in small Swiss hounds. Their research further underscores the heterogeneity of these lysosomal storage diseases, but also their shared core characteristic, progressive neurodegeneration. This particular investigation focused on 2 littermates, small Swiss hounds, who began exhibiting pronounced neurological signs around 12 months of age. The clinical picture was dire, characterized by progressive ataxia, which is a lack of voluntary coordination of muscle movements, as well as a significant loss of cognitive functions and vision. Tragically, the severity of these clinical signs necessitated euthanasia for both dogs just a few months after the onset of the disease, highlighting the rapid and devastating progression typical of many neuronal ceroid lipofuscinosis forms. Pathological examination of one of the affected dogs provided crucial corroborating evidence, revealing cerebral and cerebellar atrophy, indicating a shrinking of brain tissue in these vital areas responsible for thought, movement, and coordination. Furthermore, a definitive hallmark of neuronal ceroid lipofuscinosis, the cytoplasmic accumulation of autofluorescent material in degenerating neurons, was observed. This autofluorescent material represents the waste products that the cells are unable to process, leading to their buildup and subsequent neuronal dysfunction and death. These consistent clinical signs, combined with the characteristic histopathology, led the researchers to a tentative diagnosis of neuronal ceroid lipofuscinosis, setting the stage for their detailed genetic investigation. The pathological characteristics, including the intracellular accumulation of autofluorescent lipopigment and the loss of neurons, particularly in the cerebral and cerebellar cortices, are consistent with other forms of neuronal ceroid lipofuscinosis. Sometimes, atrophy of the retina is also observed. This study reinforces the commonalities across different forms of neuronal ceroid lipofuscinosis in dogs, demonstrating that a presumptive diagnosis can be based on evidence of heritability, progressive neurological dysfunction, and the accumulation of these autofluorescent storage materials in neurons.
Indeed, and the genetic investigation by Reitman's team provided the definitive diagnosis for the small Swiss hounds, classifying their condition as neuronal ceroid lipofuscinosis type 7, or NCL7. Using whole genome sequencing on one of the affected dogs, the researchers uncovered a significant genetic alteration, an intragenic duplication spanning 18,819 base pairs within the MFSD8 gene. This means a segment of the gene was unintentionally copied and inserted within the gene itself. The precise breakpoints of this duplication were identified in intron 3 and exon 12 of the gene. Critically, this duplication was predicted to disrupt the reading frame of the gene, meaning the cellular machinery attempting to read the genetic code to produce the MFSD8 protein would encounter a scrambled sequence after the duplication, likely resulting in a non-functional or truncated protein. The impact of such a disruption, leading to a loss of function of the MFSD8 protein, is consistent with the known mechanism of disease for NCL7. This is significant because MFSD8 loss-of-function variants are well-established causes of NCL7 in human patients, as well as in other mammalian species, including dogs. Genetic testing confirmed that both affected small Swiss hounds carried this duplication in a homozygous state, meaning they had 2 copies of the duplicated gene, one from each parent. Furthermore, there was perfect co-segregation of this genotype with the disease phenotype throughout a large pedigree of small Swiss hounds, which is a strong indicator of an autosomal recessive inheritance pattern. Both the sire and dam of the affected dogs were found to be heterozygous carriers, possessing 1 normal gene copy and 1 duplicated copy, aligning perfectly with the expectations for obligate carriers of a recessive disease. The MFSD8 duplication allele was found at a frequency of 5.7% within their study cohort of 106 small Swiss hounds, showing a complete association between having 2 copies of this duplication and the occurrence of neuronal ceroid lipofuscinosis. These robust experimental data, combined with existing knowledge about the MFSD8 gene, strongly support the conclusion that this specific intragenic MFSD8 duplication caused the disease in these small Swiss hounds, thereby refining their diagnosis to neuronal ceroid lipofuscinosis type 7. This groundbreaking discovery is crucial for the breed, as it now enables genetic testing. Such testing allows breeders to identify carriers, monitor the population, and implement targeted breeding programs to prevent further unintentional carrier-to-carrier matings, ultimately avoiding more cases of this devastating disease. This study exemplifies the power of whole genome sequencing in veterinary diagnostics, offering precision medicine approaches for inherited conditions.
These insights into NCLs are truly impactful. Now, let's pivot to a slightly different, though related, neurodegenerative disorder. Keller and colleagues, writing in Genes, describe a novel progressive neurological disorder in Weimaraners that, while sharing some similarities with NCLs, presents with unique characteristics, particularly concerning myelin abnormalities. The proband, a male-neutered Weimaraner, started showing subtle neurological signs at approximately 4 years, 10 months of age, making it a late-onset condition compared to many of the typical NCLs we've discussed so far. The clinical presentation was quite varied and progressive, including fecal incontinence, lethargy, moderate paraparesis, weakness in the hind limbs, proprioceptive pelvic limb ataxia, loss of coordination of hind limb movements due to impaired sense of body position, falling, cognitive decline, incoordination, decreased interest in food, changes in posture, and episodes of trance-like behavior. This diverse set of signs speaks to widespread neurological dysfunction. As the disease advanced, the increasing severity of these signs led to humane euthanasia at 6 years, 7 months of age. Magnetic resonance imaging of the brain revealed generalized brain atrophy and areas of white matter pathology, indicating damage to the parts of the brain responsible for transmitting nerve signals. Pathological examination further detailed the disease's distinct features. Autofluorescent intracellular granules were observed in various tissues, including the cerebral and cerebellar cortices, the optic nerve, and even the cardiac muscle. Crucially, these abnormal inclusions in the cerebral cortex and cardiac muscle showed immunolabeling with antibodies to mitochondrial ATP synthase subunit C protein, a finding consistent with that seen in the Neuronal Ceroid Lipofuscinosis group of lysosomal storage diseases. However, what sets this Weimaraner disorder apart are the pronounced abnormalities in the myelin of the brain and optic nerve, consisting of distinctive areas of ballooning between the layers of myelin. Myelin is the protective sheath around nerve fibers, essential for rapid and efficient nerve impulse transmission, so its damage severely impacts neurological function. The ultrastructural appearance of the disease-related inclusion bodies varied, and their location suggested they were at least partially derived from the myelin surrounding axons. The inclusions in cardiac muscle were also notably located in mitochondria-rich regions.
This focus on myelin is a critical distinguishing factor for the Weimaraner disorder. The genetic analysis performed by Keller and colleagues elucidated the molecular basis for these unique findings. Whole genome sequencing of the affected Weimaraner revealed a homozygous G2A missense mutation in the CNP gene. The CNP gene encodes proteins that possess 2'3-cyclic nucleotide 3-phosphodiesterase enzyme activity, often referred to as CN-PASE, and play a crucial structural role in myelin formation and maintenance. This specific mutation predicts a THR42 metamino acid sequence substitution, meaning a threonine amino acid is replaced by a methionine at position 42 in the CN-PASE protein. This substitution is predicted to be detrimental to the protein's function. To confirm the pathogenicity of this variant, archived Weimaraner DNA samples were genotyped, and another dog that was homozygous for this G2A variant was identified. This second dog also exhibited a clinical history and brain lesions strikingly similar to those of the proband, further supporting the causal relationship. A broad survey confirmed the specificity of this finding. Out of 304 Weimaraners and over 4,000 other dogs from various breeds, only the 2 affected Weimaraners were homozygous for this specific CNP missense variant, providing strong evidence for its direct link to the disease. Further investigation into the protein itself revealed that while CN-PASE immunolabeling was widespread in the brain tissues of normal dogs, it was undetectable in the same tissues from the proband, indicating a CN-PASE deficiency as a direct consequence of the mutation. This deficiency, in turn, is directly implicated in the observed myelin abnormalities. The authors conclude that based on the comprehensive evidence, including the clinical history, fluorescence and electron microscopy, immunohistochemistry, and the molecular genetic findings, this late-onset Weimaraner disorder most likely results from this CNP missense mutation. This leads to the CN-PASE deficiency, which then causes the myelin abnormalities, the accumulation of lysosomal storage bodies, and the observed brain atrophy. Interestingly, similar disorders have been associated with different CNP variants in other breeds, such as Dalmatians, where a homozygous nullifying single-base deletion and reading frameshift in CNP led to slowly progressing neurodegenerative signs. Some relatives of these Dalmatians, who were heterozygous for the CNP deletion, even began showing neurological signs later in life. These findings underscore the critical role of the CNP gene and its encoded protein in maintaining neurological health and myelin integrity across species, offering another valuable canine model for understanding human myelin-related neuropathies.
That connection to myelin is truly a unique aspect of the Weimaraner case, distinguishing it within the broader spectrum of neurodegenerative diseases. Now, let's return to a more classic neuronal ceroid lipofuscinosis, specifically type 6, as described by Mlangamutangudura and colleagues in Genes, this time in a mixed-breed dog. This study reinforces the pervasive nature of neuronal ceroid lipofuscinoses as among the most common hereditary neurodegenerative diseases in dogs. As we've established, these are lysosomal storage disorders characterized by neurological signs that progressively worsen over time, leading to central nervous system atrophy and the distinctive autofluorescence of the lysosomal storage bodies. While mutations in at least 13 genes have been associated with various forms of neuronal ceroid lipofuscinosis in human subjects, canine orthologs, the corresponding genes in dogs have been implicated in the majority of these conditions. However, it's also important to note that other hereditary disorders in dogs with NCL-like phenotypes have been described, resulting from mutations in genes not yet directly associated with neuronal ceroid lipofuscinosis in humans, such as the ARESG gene and the CNP gene we just discussed. The case presented in this paper involved a 23-month-old neutered male, mixed-breed dog that had a 4-month history of rapidly progressive neurological signs. These signs were tragically comprehensive and included anxiety, cognitive impairment, tremors, seizure activity, ataxia, incoordination, and pronounced visual impairment. The severity of the brain degeneration was evident on magnetic resonance imaging, which showed marked diffuse brain atrophy. Due to the relentless progression of these debilitating neurological signs, the dog was humanely euthanized at approximately 26 months of age. This case highlights the rapid and severe progression of these disorders when left untreated, emphasizing the urgent need for early diagnosis and intervention. The pathological examination confirmed the presence of the characteristic autofluorescent lipofuscin-like inclusions, which are the hallmark of these storage disorders.
And indeed, the pathological findings in this mixed-breed dog with neuronal ceroid lipofuscinosis type 6 were quite telling, providing strong evidence for the diagnosis. Post-mortem examinations revealed the presence of the characteristic autofluorescent lipofuscin-like inclusions in the retina, cerebral cortex, cerebellar cortex, and cardiac muscle. The detection of these autofluorescent granules is a critical diagnostic feature for neuronal ceroid lipofuscinosis. Further immunohistochemical staining demonstrated that these inclusions were immunolabeled with antibodies to mitochondrial ADP synthase subunit C protein. This finding is highly significant because the storage of subunit C of mitochondrial ADP synthase is a fundamental biochemical hallmark of many forms of neuronal ceroid lipofuscinosis, particularly those affecting the CLN6 gene, providing strong evidence of the underlying biochemical defect. The researchers also observed pronounced neuroinflammation within the brain tissues, indicated by significant astrocyte activation, immunolabeling for glial fibrillary acidic protein, or GFAP, and microglial activation, immunolabeling for ionized calcium-binding adapter molecule 1, or IBA1. This neuroinflammation is a common secondary pathological process in many neurodegenerative diseases, representing the brain's attempt to respond to injury and cellular dysfunction. The genetic investigation confirmed a splice site variant in the CLN6 gene. A splice site variant affects the process by which messenger RNA is correctly processed from a gene, often leading to errors in protein production. The identification of this specific CLN6 variant in a mixed-breed dog, along with the characteristic clinical signs and pathological findings, reinforces the understanding of CLN6-related neuronal ceroid lipofuscinosis. The results are consistent with previous reports of CLN6 mutations causing neuronal ceroid lipofuscinosis in other breeds, such as the Schapendoes we discussed earlier and Australian Shepherds. These findings continue to expand our knowledge base of the genetic causes of neuronal ceroid lipofuscinosis in dogs and highlight the importance of genetic screening, even in mixed-breed populations, for accurate diagnosis and genetic counseling. While this dog was of unknown ancestry, the genetic identification of a CLN6 variant clearly places its neurodegenerative disorder within the well-defined framework of neuronal ceroid lipofuscinosis type 6, demonstrating the unifying molecular basis of these diverse clinical presentations. The consistency of the findings with the known pathology of neuronal ceroid lipofuscinosis, such as the subunit C protein accumulation and the autofluorescent inclusions, strongly supports the diagnosis and the role of the CLN6 variant in this dog's condition.
Fascinating how these cases, though in different breeds or even mixed-breed dogs, often converge on similar pathological mechanisms. Our final paper, by Guo and colleagues, published in Genes, explores a distinct neurodegenerative disorder in Nova Scotia duck tolling retrievers, classified as degenerative encephalopathy, or D. This is a slowly progressive, inherited neurological condition, and it presents with a particularly striking and consistent clinical sign: frequent episodes of pronounced involuntary movements during sleep. This unique manifestation sets it apart from some other neurodegenerative diseases we've discussed. Beyond the sleep disturbances, affected Nova Scotia duck tolling retrievers also exhibit a range of other debilitating symptoms, including cognitive impairment, anxiety, heightened sensitivity to sensory stimuli, and compulsive behaviors. As the disease progresses, some affected dogs develop additional signs such as aggressive behavior, gait abnormalities, and urinary and fecal incontinence. The severity of these signs increases over time, profoundly impacting the dog's quality of life. The onset of these symptoms can vary widely, reported to occur as early as 2 months of age and as late as 5 years. Brain degeneration accompanies these clinical signs, indicating a progressive assault on the central nervous system. The research aimed to identify the molecular genetic basis of this specific disorder, utilizing whole genome sequencing on affected and unaffected Nova Scotia duck tolling retrievers, alongside control dogs from other breeds. This comprehensive approach is essential for isolating the specific genetic variants responsible for such complex inherited conditions. The identification of such a variant is crucial for developing tools to manage and eventually eradicate this debilitating condition within the breed population.
And the genetic findings in the Nova Scotia duck tolling retrievers were indeed conclusive, providing a clear understanding of the underlying cause of their degenerative encephalopathy. Whole genome sequencing revealed a missense CTT variant in exon 22 of the RB1CC1 gene, located on chromosome 29 at position 4891014. This variant was found to be uniquely homozygous in the affected dog, meaning the affected individual had inherited 2 copies of this specific mutated gene, one from each parent. This genetic alteration predicts a P.G1503R change in the amino acid sequence of the RB1CC1 protein, where a glycine is replaced by an arginine at position 1503. Such a missense mutation can significantly alter protein function. The strength of this finding was further solidified by comprehensive genotyping of over 2,900 Nova Scotia duck tolling retrievers at this specific variant locus. This extensive analysis found complete concordance between the disease phenotype and the RB1CC1 genotype, providing compelling evidence that this variant is indeed the cause of the disorder. The discussion section of the paper highlights the critical role of the RB1CC1 gene. RB1CC1 is known to be an essential component of a protein complex that mediates the initiation of autophagosome formation. Autophagy is a vital cellular process responsible for degrading and recycling damaged cellular components and organelles. Therefore, the researchers concluded that it is highly probable the degenerative encephalopathy in these dogs results, at least in part, from impaired autophagy. Consistent with this proposed mechanism, brain neurons of an affected dog were observed to contain abnormal lysosomal storage body-like inclusions, which further supports the idea that the cellular waste disposal system is compromised. This is similar to how lysosomal storage diseases lead to the accumulation of abnormal materials, even though the primary defect is in autophagy rather than a specific lysosomal enzyme. When damaged intracellular components cannot traverse the autophagosome to autolysosome degradative pathway, they appear to be sequestered in these inclusion bodies. While not classified as a typical neuronal ceroid lipofuscinosis, this disorder presents with NCL-like pathological features due to the accumulation of lipofuscin-like inclusions, which is a known consequence of impaired autophagy. This study's findings are particularly significant because variants in autophagy genes have been associated with a number of human autosomal recessive hereditary neurodegenerative diseases, including forms of ataxia, intellectual disability, seizures, and even schizophrenia. Therefore, this canine disorder in Nova Scotia duck tolling retrievers can serve as a valuable model to elucidate the mechanisms underlying human diseases associated with impaired autophagy. The identification of this specific disease-causing DNA sequence variant is an immense practical benefit for owners, enabling them to screen their dogs for the RB1CC1 risk variant and implement responsible breeding practices to avoid producing affected offspring.
These collective studies truly highlight the complex and multifaceted nature of neurodegenerative diseases in dogs, ranging from well-characterized neuronal ceroid lipofuscinosis caused by specific lysosomal enzyme deficiencies to myelin abnormalities and impaired autophagy. The ability to identify these specific genetic causes, often through whole genome sequencing, provides not only definitive diagnoses, but also crucial tools for breeders to manage and prevent these devastating conditions. Furthermore, the striking similarities between canine and human neurodegenerative disorders position dogs as invaluable natural models for research, accelerating our understanding of disease mechanisms and the development of potential therapies that could benefit both canine and human patients. The progressive nature of these diseases, their early onset in many cases, and their often fatal outcomes underscore the importance of continued research in this area to improve the lives of affected animals and, by extension, contribute to human health. Each of these papers adds another piece to the intricate puzzle of inherited neurodegeneration, from pinpointing specific genetic variants to unraveling the molecular pathways, such as myelin synthesis or autophagy, that go awry. The commitment of researchers to investigate these rare but impactful conditions offers hope for better diagnostics, genetic counseling, and ultimately, effective treatments.
Thank you for listening to the Veterinary Medicine Digest. If you're enjoying this, remember to like, share, and subscribe, as proceeds from this channel go directly to providing vital veterinary care for animals in need. Even a small amount can transform an animal's life and ease an owner's burden. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.